what spurs this discussion and this document that I prepared. So if y'all looked at the document, Reducing Biotech to Practice, what spurs this and me talking about this is a couple of things. One, I feel like some people were having some issues with the projects, figuring out what to do next or where they should go. They have their idea, but they might not know what to do next. So I think this document is going to add a little bit of clarity to that. And then I also had another student come talk to me about this issue in particular, about learning a lot. I've been teaching a lot of techniques and teaching a lot of language and teaching a lot of like things and items, but a question is, okay, well, how can I actually like use that now? What's kind of like the thought process? How do I put this stuff into practice? So I want to address that comment and that criticism, and I want to try to do my best to walk through how you can reduce the things that I've been teaching to practice. So I just want to walk through this document. Let's walk through it in Photoshop. So let me zoom in here. Okay, so anytime you're working, I guess, in science, biotechnology, anything related to science or biology at all, the first thing you need is you need some kind of a question, okay? What's your big question? Or in terms of biotech, maybe that gets phrased as what problem are you going to solve, okay? So you need to start there. If you don't even have like a question or an idea, you can't start past that, okay? So you have to get that. If you don't have an idea, if you don't have an idea, okay, go back, watch that lecture that on getting good ideas, and I would suggest that you journal for 10 minutes a day until you get better ideas. And you'll slowly sort of build up the mental infrastructure to have ideas kind of like spontaneously come to you. And now that you guys are loaded with kind of the knowledge and the language, you're going to start to have ideas about, oh, I should do this, because you're starting to learn the language and the techniques. Okay. So if you don't have an idea, journal. Imagine you do have an idea. Okay. Most of you in this class now, we're at the point where we do have ideas. So you know what you're asking. Okay. So you move this way. Has it ever been done before? So what this would mean is the first thing you need to do is you need to go to Google. You need to Google and check that. Okay. Because the worst thing is putting in massive amounts of investment of time into doing all the hard scientific work. And then you find out a year or two later that somebody has either already done it completely or somebody's like competing with you and doing it at the same time. So go to Google first thing, Google your idea and try to phrase it in as many different writings or languages, not languages, but like terms. Try to phrase your idea in many different ways and see if you can find anything on the subject, okay? So if it's been done before, you got to go back, you got to think of a new question. And I would also say, in addition to Google, you're going to want to check NCBI. So one of the search things you can search for is publications. So just like before, I showed you how to search for genes or proteins, you can search for publications. Can you find any publications? You want to check Google Scholar, the patent database. So if you just Google US patent database, you will come up with the US patent database website, and you can Google in that database just like you would in NCBI or just like you would in Google. And that's another great resource. And interestingly, in the patent database, what's published in there is not peer reviewed. So you're going to find a lot of what people are working on like very, very recently in the patent database. So search these three things to determine if your idea has already been explored. If yes, get a new idea. If not, then you're good to go. So if not, that's your best case scenario. Now, the next thing you go to is this question, I have a desired phenotype in mind, okay? That means like whatever problem you're going to solve or whatever question you're addressing, you cannot study anything or work with anything if there's nothing to observe or measure or There's no phenotype, okay? So you need some kind of a phenotype. It's better if your phenotype is easy to see, easy to measure. And a lot of research is literally just like figuring out the best way to measure certain phenotypes, okay? Because it's an extremely important part of research. If you don't have a phenotype, you have nothing to work off. You have nothing to measure. So the next step is you have a phenotype in mind. Okay. So imagine the answer to this question is yes. Okay. You have a problem. It hasn't been done before. You have a phenotype in mind. Okay. Now there's three potential options. One option is you know the phenotype, but you know the genotype. Okay. That means you know the genes that are causing the particular phenotype. If that's true, that's fantastic. Okay. The other scenario is you have a phenotype in mind, you know the phenotype you're thinking about, but you don't know the genotype. That means you don't know the genes, you don't know what the genes are that are causing or responsible for that phenotype. Okay. These are two different research trajectories, and it's going to drastically influence the things that you are trying to do, depending on whether you know the genes that cause the phenotype or not. Okay. Hopefully, eventually, 10,000 years from now, we'll get to the point where we know the genotype for every phenotype, but we're probably a little early at this point. The third option would be that you're thinking about something that doesn't involve genes. Okay. That could still be related to biotechnology, but if it's something that's not involving genes, you probably don't have the background knowledge to work with it. So I suggest if you're like thinking about something like chemical engineering, right? Like learn pharmacology, learn biochemistry. If it's something related to like informatics or something, it might be you're going to need to learn computer science, or you're going to hire other people to do it or collaborate with other people. Okay. So if it doesn't involve genes, you need to learn and expand yourself into new fields. If it does, then you kind of flow either this way or this way. Okay. So let's tack on this trajectory. So you don't know the genotype. You don't know the genotype that causes the phenotype of the particular thing that you want to create. Okay. The next thing you do, if that's the case, is you need to run a screen, some type of a screen. Okay. The screen is an idea where you have, you may have 1,000 or 10,000 different variables in play, and you need to figure out which are the ones that are important, right? Which are the genes that are important for causing that phenotype. And to do a screen, you need some kind of a readout. Okay. You need some kind of something to look at. Usually that's going to be your phenotype. So let me talk about some different types of like screens that if you were going down this research trajectory, some of the things you could do. Okay. And we should now have a good understanding of what these things are. So a lot of people just sequence stuff. That's kind of like the first thing. So imagine your question is, I want to study phenotype X in what's a very strange organism that hasn't had a genome sequenced, in a weird tree, a weird babinga tree. I just say this as an example of something that probably doesn't have its genome sequenced. There's probably no genome. Okay. And if you don't have a published genome, that's a huge like research limitation right there. And it's highly unlikely that you're ever going to find the cause of phenotype X in a weird babinga tree if there's no genome. So that's kind of like one of the first things you can rectify. Okay. So sequence stuff, get a genome. Got it. Next generation sequencing. So that would be like Illumina, HiSeq. You can also do RNA-seq. You kind of need a genome for this, but not necessarily. Okay. You could sort of get hits based off BLAST. But the other thing you could look at is like, what is it expressing? Get like a transcriptome or a genome. At least then you know kind of like what is the repertoire of things that are being expressed in weird babinga tree, etc. Okay. That's like starting point one. Okay, more about screens. The next thing that most people commonly do is they start breaking stuff, okay? And this is, again, assuming you don't know the gene you want to target. You don't know what it is. You don't have a target. So in the case where you don't have a target, you need to sort of target everything in the genome. So utilizing things that integrate or break stuff randomly is very useful. And I made the analogy before where if you have like a shotgun, okay, and you just like fire a shotgun at the genome of a particular organism and you cause random mutations. Mutations give rise to X phenotype, which you are looking for. Then you can sort of trace back, where is that mutation? What gene is it in? That is probably important for X phenotype, okay? So there's many different ways to do this. And I'm listing a few here, okay? One is a transposon insertion screen. So I talked about this in class in reference to one student's project where imagine, again, you're trying to find some phenotype or the cause of some phenotype. You can make a plasmid with a helper and a donor. And in the donor, you just put some kind of selectable marker with IRs, inverted repeats, or transposase recognition sites. Helper plasmid is going to be some kind of transposase. Okay. And you can this can be your shotgun. Let these two plasmids be your shotgun. And what they're going to do is you put these in the cell or the organism. It's going to make the transposase. It's going to grab the selectable marker, and it's going to pop it in randomly, shooting the shotgun at the genome. Okay. That's one way to do that. It's a very, very common thing that people do. Another variant on this, but the same idea, is you can do recombinant protein expression of the transposase. Okay, so you make a whole bunch of T, and you can get it pre-bound to your marker. So in this case, you'd be sort of like injecting plasmids. You can sort of cut a step out, and you can, well, it would actually add a little bit more work, but you can do recombinant protein expression, purify the actual transposase protein, complex it with this part, and then you can microinject or transform this protein DNA complex. And this is going to have a very, very high efficiency. So it'd be, imagine like a shotgun with like many, many, many little beads. This is going to have a lower efficiency. A shotgun with less beads. Okay. So there's different ways to do this. Okay, we've also talked about agrobacterium, the Tdna insertion screen. Okay, that is, again, the same idea, except this case, your helper is your endogenous Ti plasmid, which expresses, I think they're over here, the Vir proteins. And your donor would have the Tdna region flanked by the sites that the Vir proteins recognize with some selectable marker, okay? And then you can shotgun in kanamycin cassettes with agrobacterium in plants or whatever organism you want to use that for. I did not want to click that button. No, please don't do this. Okay. The next thing you could do is a CRISPR knockout screen. So again, these are all different ways that we can break stuff. And I'm explaining each one of these. Okay. A CRISPR knockout screen, similar idea. You're engineering a shotgun. The way that you would do it is Cas9. It's kind of dangerous getting random emails while I'm recording. Hopefully nothing controversial comes in. Okay. You can, okay, a Cas9 knockout screen. What would this look like? So Cas9 is the nuclease. Again, we haven't got to CRISPR yet, but we'll get there soon enough. Cas9 is a nuclease. We've talked about it enough in the context, you know, that it cuts DNA, it induces double-strand breaks, and the specificity of it is regulated by the guide RNA, okay? And you can create libraries of different guide RNAs. And imagine you just had random guide RNAs. A library of random guide RNAs is going to cut the genome, shotgun the genome at random sites, okay? So if you design your guide RNAs with some kind of random algorithm, you can randomize the Cas9 cutting and use it as a shotgun. Okay, so that would be like a CRISPR knockout screen. You could also, if somebody has already worked and done all these things. So imagine if you needed to make it from scratch, you could do 1, 2, or 3. If you're working in something like yeast or E. coli, there's probably already a deletion library, which means that if you're working in a model organism where there's been so much study done, it's probably true. It's definitely true in Saccharomyces cerevisiae that there's already a library of strains where they essentially go through all the different genes and knock them out individually. And there's a clone for each of these knockouts. So you can do experiments with knockout strains looking for your phenotype X, okay? This would be using what would be called a deletion library. And this is one reason why yeast is so effective, is because there's already a whole bunch of rigorous deletion libraries already done. You don't have to go through the work of engineering the shotgun. And then we've kind of talked a little bit about directed evolution. Directed evolution would be the scenario where either you're mutagenizing with some kind of like a chemical. You could add like a chemical or like a UV light. You're inducing damage to the DNA, and you're having some kind of like passage system where you're progressively trying to passage something towards some end state. So you're providing a little bit of mutation, and you're trying to get the organism to go from X to Y. And once it gets to Y, you could then like sequence its genome and figure out like what changed. That would give you then your list of what causes the Y end state. Okay, but this would imply you have some way to select it, culture it, and sort of like push it towards this end state. You may or may not have that. Something in the chat. With CRISPR, would you be building a library of guide RNAs to perform loss of function screening? Yes, that is exactly what I was saying. You're building a library of guide RNAs. And in this case, I said that they were random. They're literally just random sequence, ATGC, whatever. And if they're random, they're going to target randomly in the genome. You could make a more targeted like library if you wanted. But that defeats the purpose of what we're talking about here, because we're talking about how to engineer random shotguns at the genome. Okay, so again, the issue of this tree is you have a phenotype in mind, you don't know the genotype, you want to try to find all the genes that are responsible for the phenotype. So you can sequence stuff, you can break stuff, you can make stuff. So this is the reciprocal of the break stuff. And I talked about this in the lab, where up above, all those things above are loss of function experiments. You're doing some kind of situation to create loss of function. The inverse of this is gain of function. Okay, you're doing some kind of screen or library to induce a gain of function. So let me talk about some of these. Okay, 1, Overexpression Library. There's so many things open. Overexpression library, what would that be? Okay, so for instance, in yeast, if you take a 2-micron plasmid, that's high copy, the high copy plasmid, and all you need to do is clone chunks. Oh my gosh. I'm gonna have to like shut off my email. I should just close it. But if I open it up, you'll see everything that's in there. Okay, there we go. Close my email. Okay, clone chunks of yeast genome into a high copy expression plasmid. So imagine this is the full yeast genome here to here. You can cut it into chunks with some restriction enzyme. So you got chunk 1, 2, 3, 4, 5, 6, 7, on and on and on and on. And you clone these chunks, and in these chunks is probably multiple genes. Probably got a few genes. Some going that way, some going this way. They all have their own promoters. They all have their endogenous promoters. Okay. You clone these chunks into the high copy plasmid. Now the high copy plasmid, when it replicates, there's about 60 copies of it. So now you've created cells that are aneuploid, probably spelled that wrong, aneuploid for genomic segment 1, okay? So now you have a situation where whatever these genes are from that chunk, you have 60 copies of those genes, okay? So they're all gonna express at sort of like greater than 60-fold normal values, okay? They're gonna be overexpressed. Even if you're not inducing expression, or even if you're not engineering like regulation of the promoters, they all have their own promoters. And just the fact that there's 60 more copies of them in that particular cell, they're going to be expressed at 60-fold higher rates. Okay. So this would be an overexpression library. Now you can imagine if you have a phenotype that you want to study, imagine you are working in a cell and you have phenotype X. Imagine, this is the example that I give in my CI lectures, where we had a gene called SIDB. SIDB was a toxin. So the phenotype was that it kills the cells. It kills them, okay? And then what we did is a suppression screen with a high copy overexpression library in yeast. So in this cell, there's the SIDB toxin on a plasmid, and then we transform it with 1 to N high copy plasmids. And if there is some gene Y, let's call this one Y, that inhibits the SIDB toxicity or the toxin, then these cells will survive. And so if you do this, you take these cells, you transform them with the whole library, and then you create plates. The only ones that are going to grow are the ones that have what we call a suppressor. It's suppressing some phenotype. In this case, the phenotype is a toxin that kills the cells. If you can identify suppressors of the phenotypes, you probably are pointing to what pathway the particular gene is involved with, getting closer and closer to determining the genotype that determines the phenotype, et cetera. Okay, so this is what we call a suppression screen. Okay, number 2, recombinate large genomic chunks. That's kind of what I've already explained here, where if you're doing gain of function experiments, you can create scenarios where you're cloning out large chunks of the genome that might have large, essentially like a whole bunch of ORFs. And this is on a plasmid in this case. So it's a smaller way to do it. You could do this at an even larger scale if you're working with BACs, which are bacterial artificial chromosomes, or YACs, which are yeast artificial chromosomes. You could clone massive sequence, 120 kb, into a YAC backbone, and then you can transform this YAC backbone into some cell and see if it produces some kind of gain of function. And then from there, you can work backwards, where maybe you would cut this piece in half, make 2 different yaks, yak 2, yak 1, transform these into cells, look for that gain of function, and if it's in this one, then you know that it's in this part of the genome, and then you just progressively get smaller and smaller and smaller and smaller, till you can find like maybe like an island of orphs that's causing something. Okay. And then directed evolution again. So essentially everything I've covered here now there's a whole bunch of ways you can break stuff randomly. There's a whole bunch of ways that you can make stuff through gain of function experiments. And then a final type of screen, which I have used, which has worked very well, is if you have, say, a particular protein, you might be able to figure out like what does it bind to. Okay. Oftentimes in cells, if you have protein X does not operate in a void. Okay. It doesn't operate like by itself. Proteins are kind of like always in interactions with other entities, right? And if you can start to find kind of like what are the friends, this is colloquial language, what are the friends or what are the networks that your protein X interacts with, almost like the Facebook page and the friends list of your interesting protein X, then you can start figuring out like what pathways it's involved with. Like for instance, the CIA might do this if they're looking at individual X and they notice that X is always interacting with Y terrorist and A terrorist, then you know that X might possibly be a terrorist, right? So it's kind of like the same rationale where in the cell, if you can start finding the friends or the networks or their interactomes of X, and if you might not know the function of X, but you might know the function of A, that's going to give you an idea of what X is doing, what it's working with. So identifying like an interaction network is a way to do that. And to do that, you can do a pull down. So you could set up a cobalt column. This is cobalt. And imagine you have His6 protein X, and you want to identify all the A's and the Y's and all the other proteins that it's interacting with. You could grow some bacteria in a culture flask, or if it's a fly protein, you grow some flies, or if it's a mosquito or a plant, you grow some plants, smash them up, and you run them over the column. And if protein X interacts with protein A, this entity is going to get stuck to the column, okay? And then you go through a series of washes. So I wash this with buffer. I just progressively wash it, and stuff drips out. And I'm washing and washing and washing and washing. And the only things that are going to remain are protein X, because it's bound to the cobalt column with the His6, and whatever it likes to bind to really, really tightly. That's going to remain, okay? And then once you've washed it, you can do an elution, and you can figure out what all these entities are that it's interacting with through liquid chromatography mass spectrometry, tandem mass spectrometry. And that's in lecture 3 if you don't remember what that is, or you could YouTube it or Google it. It's a way to figure out what is A. Question in the chat. How could you determine whether loss of function or gain of function would work best in a given experiment? It's going to depend on the phenotype. Okay, so the question, let me type it out, or I'll write it out. How to know if loss of function versus gain of function experimentation is the better approach. Like, what's the better approach? It's definitely going to depend on the phenotype, okay? For instance, imagine your phenotype. Let's go back to the Hox example. So the Hox gene example was what genes, actually, this is reciprocal of what I wanted to say. Okay. Let's imagine you have a fly, okay? a normal fly, 2 wings, and you want to know what genes control wing formation. Okay. If you shoot a shotgun at it, so that's going to be the loss of function style, and you yield a fly that has no wings, then that's a good way to do it. And you can identify the genes that are responsible for that. The reciprocal would be like you could do the same kind of thing. And if you get the fly that has the 4 wings, I don't know if you'd consider that a gain of function. It's probably not a good gain of function. But do you see how it's the phenotype that you're looking at that's determining like what you do? So for instance, me, in my example, in the yeast, I have the plasmid, the SID-B plasmid, which is a toxin, and its phenotype, again, it kills the yeast. Okay, that's the phenotype. So this is kind of like a negative phenotype. They like die. They don't grow if this gene is there. And I want to find things that suppress it. So this is more of like a gain of function type thing. Like you're gaining the ability to suppress the way that you draw this is you're gaining the ability to inhibit the toxin. So that's more of like a gain of function type thing. So it's going to depend on the phenotype. And it's going to just think about like, am I breaking something or am I gaining a function? And what's the readout of the phenotype? Again, you have to like look at the chart. You have to look at your chart. Your readout is your phenotype. That's going to determine like what you can do. And nobody can sort of like figure that out for you. It's your job. But these are some things you can think about. Okay, so we've gone this trajectory on the right. Let's go this trajectory on the left, which is I have a desired phenotype in mind. I do know the genotype. This means this would be, for example, like we know the genes that turn on fruit fly wings. We already know that. We know what the genes are. Okay, so we know the genes that control the phenotype. Okay, and then your options are you going to need to work with like a couple genes, a few genes, like 1 to 3 to 4, or are you going to need to work with like a whole pathway of like 25 genes, like 100 genes, 1,000 genes? Okay, if it's too many, the phenotype is going to be simply like too complex. You're going to have to find like a simpler model, or you're going to have to somehow reduce the complexity by sort of like working through this list. You need to somehow reduce the complexity. There's only like so much you can do, unless you're like really good. Like if you're really good, there's ways to like think through it. What is too many? Yeah, like that, like that. See, I mean, it depends on your ambitions, right? Like there's people who are literally rewriting and this is actually funny for the last lecture. There are people who are quote unquote intelligently designing the yeast chromosome. So yeast is such a well-loved model, and it's such a well-loved eukaryotic experimental system that there are people who are taking the yeast chromosome and they're essentially like deleting all the things from it that make it inconvenient. Okay, like virus genes, transposons, essentially like getting rid of all the trash, and they're like reorganizing it in a way where it's kind of like organized in a way where they're like building operons, making things like localized together so that it makes sense. And then they're also like adding in features, adding in things you might want, like recombination sites or things like that. So these people who are doing this, intelligently redesigning the yeast chromosome, they are working with 5,000 genes. I mean, and there are people who are able to do this. Okay, but if you're not one of those people and you're not there yet, I'm essentially saying to you that like you might want to reduce your complexity. But I would also say like the sky's the limit. Like, you know, you can do whatever you want. You could there's people who have built an entire new organism. The mycoplasma, which I talk about, the mycoplasma the minimal bacterial mycoplasma strain, they literally assembled that. That was not on the Earth. That was not on the planet before they built it, and they built it. And that was 300 genes. So you're starting to hit some real complexity when you're working with 300 genes to 5,000 genes. If you can't handle that, I would suggest starting with 1 to 2. Definitely start with like 1 and then get comfortable with like working with 1 gene and then maybe like move up. Okay, but imagine you're working with a few genes. Okay. And then usually the next thing you can do is, okay, these are scenarios where you want to produce a protein or a chemical. Like I want to produce psilocybin, which is going to be the second to next lecture. So I need the enzymes in the pathway to synthesize that. That's 4 genes. Okay. Or imagine the next lecture, which is I want to produce spider silk. That's 1 protein. So you only need like 1 gene. Okay. Usually in scenarios where you're doing something like that, I want to produce spider silk. I want to produce psilocybin. I want to produce insulin, etc. Usually you're cloning like a gene onto a plasmid. That's the first thing you do. Okay. So you clone your gene X that makes product or protein X. You clone that into a plasmid. Usually you're going to do that with restriction enzyme cloning or some of the other methods that we talked about. You're going to run some agarose gels to make sure you got it, and you're going to Sanger sequence it to make sure that it's perfect. Okay. From there, there's now a question. Do you know how the gene works? So if you're working with insulin, people already know what insulin does. They already know its functions. They already know what it does. Spider silk, we kind of like know what its function is. We kind of know what it does. If you're cloning a gene for the purpose of figuring out what it does, then the answer would be no. So in my case, in my example, and this will come up in the CI lecture, when I cloned originally first the SIDB gene, we had no idea what it did. Okay. Like we have no idea what it did. We just knew, we identified it from a screen. So imagine initially my pipeline of workflow would have gone like this. I knew the phenotype. There's so much green on this now. Let me see if I can get rid of this. There we go. In my situation, I had a phenotype I was studying, CI, and we did not know the genotype. So I ran some screens and I found a protein that we thought was responsible. So then once I found the genotype, I could segue to this section of it, okay? Then we cloned it to produce the protein to start testing it, but we didn't know how it worked. So we went down this trajectory, okay? So if you have a protein, you know it's important for a phenotype, but you don't know what it does, now you go down this pathway and you can start doing some basic things, right? Like you can do some biochemistry experiments. So if you look at if you were to look at the gene X in the conserved domain database and it translates into protein X, and if maybe protein X is annotated to have some particular domain, and we know what this domain does, that's good. That's like a hint. Now maybe you explore that and prove it. So maybe if it's like a phosphatase, or if it's a ubiquitinase, or if it's a deubiquitinase, or if it's a kinase, like that's going to give you some hints for what it's doing, and you can explore that with biochemistry. Okay. But to do that, you're going to have to recombinant express the protein. You're going to have to express it. You're going to have to do some SDS page gels to make sure that it's being expressed. Probably you're going to have to do some Westerns to either figure out where it's localizing or make sure that you're expressing the right thing. Maybe you'll do some mass spectrometry to identify like what it's interacting with. Maybe you'll do some X-ray crystallography or some cryo-EM to figure out what the structure looks like. Then from the structure, maybe you can determine what its function is, because oftentimes structure determines function. These are some basic things you can start doing. Okay. Another common approach is things that deal with localization. So in the very, very, very first lecture, I talked about how cells are like geographical entities, okay? And they have these like distinguished spaces, and organelles and eukaryotes are cells with things in them. And if you can figure out where a particular protein is, like imagine you find your protein in the nucleus, You probably know it's probably doing something that interacts with DNA, okay? Because that's where the DNA is. That's the purpose of things that are in the nucleus. They usually interact with the DNA. So you can learn a lot about something based on where you find it. If you find it in the mitochondria, you probably know that it's probably doing something for energy or metabolism, okay? So you can learn a lot from a protein based on where it is. So a lot of people will do localization. So you could do fish to localize the messenger RNA. You could do immunofluorescence to localize the protein. You could fuse it to GFP to figure out where it's going in live cells. So this is going to be live. This is going to be dead. This is going to be protein. This will be protein. This would be messenger RNA. And you can do microscopy then to see where things are. Okay, that's another common trajectory. And then finally, if you have your cloned gene on a plasmid, so for me, right, we had our cloned gene on a plasmid, we started running some biochemical tests to explore what domain does. And then once we did that, then we started breaking it. We started shooting mutations into the SIDB gene, into our gene that we want to study, gene X, and you see if you can break that functionality by mutagenesis. Then that sort of gives you a good confirmation of what it's doing. So you can do site-directed mutagenesis, you can do SLIMs, you can do error-prone PCRs. See how I'm really only doing those like nucleic acid techniques when I'm really like far down in the ladder of thinking, okay? So again, another problem that people are encountering with their projects is they're like saying, oh, I'm gonna do SLIM, or oh, I'm gonna do site-directed mutagenesis. And they're like up here. That is not gonna work, okay? You need to like be down here in the flow chart to even have those things make sense. Okay, so this was the trajectory where you have a gene that causes a phenotype, you know what it is, but you don't know how it functions. Imagine you do know how it functions. That's this trajectory, okay? Then essentially you're at the point where you're at the last sort of phase of biotechnology where you're probably at the point where you want to distill and install that function in a new organism. So this would be the scenario where you have this gene that causes spider silk, you know what it does, you want to give another organism the ability to produce spider silk. This is like where you're at now here, okay? And then it's like, which kind of thing are you working with? Are you working with a plant, an animal, or a single cell? So like fungi would probably be like a single cell at some points in their life stage, although that's certainly not true for like fruiting body and stuff. So I've sort of ignored fungi here. Please forgive me. But if you're working in a plant, you could do like aggro to insert it. You could use gene gun to insert it. Or if you're working in an animal or a cell culture, or bacterial cells, you're probably going to be doing some kind of a plasmid, or you can use transposons to insert things. You can use viruses to insert things. You can use recombination to insert things. You can use CRISPR-Cas9 to insert things, or you can just do things the old way and you can breed stuff. So this is kind of like a framework of when you're thinking about your project and when you're thinking about like how to approach some of your questions, start thinking through this flowchart, and hopefully this will organize your thinking a little bit better. Now, I will finally just talk about this little box. This is just a helpful box to make sure you thought of all the things that you need to think about to make sure that your experiment is going to work. Okay, so all things considered, make sure you think in detail about the organism that you're working on. What strain are you working with? Is the strain optimal for whatever your particular application is? Or would it be better to work in some kind of a model? Is it better to work in a mosquito, or is it better to work in a model fruit fly? Like only you can decide, and there are certain situations where deciding to work in a mosquito would be a horrible idea when you could do the same experiment in a fruit fly. Okay, so you really need to consider like which organism are you working with, and like just to hammer this, people usually get like when people join graduate school, they get put with some PI, primary investigator, who's like their mentor, and their mentor kind of has like a repertoire of things they work with. Like maybe like I am a bad example because I work with so many different things, but maybe you work with somebody and they study like they study like a certain fish, like they study like catfish. And they're kind of like, that's what we do. We study catfish. And so you are forced to think of like, oh, everything must be done in the catfish system. That is not true, okay? There might be a better model fish, okay, that is not catfish. Maybe it's a goldfish, maybe it's a carp, I don't know, I'm just making stuff up. Maybe it's a fruit fly, maybe it's a yeast cell. Maybe you can do a particular experiment a lot faster and a lot easier in a model first before going to like a catfish, okay? So you need to think about that. Are you picking the right plasmid? There's many different types of plasmids. Are you picking the right vector? Many different types of vectors, many different types of viruses. Do you have the right selection? Is your selection going to work? Okay. Do you have selection for all the organisms that you need to work with? In many of the plasmids that we're looking at, we have to culture them in bacteria, and then we need to transfer them to a fruit fly. And so you need two different selectable markers, one for the fruit fly, one for the E. coli, et cetera. Okay. Do you have a reporter or a marker? Okay. Many times you need some kind of a readout to see if an insertion has worked or not. Okay. So do you have the proper reporter? Do you have the proper marker? Is it the right one for your particular organism? Does it work well, et cetera? Regulation and promoters. Do you have the right promoters installed? Do you have the right regulatory networks installed? Do you need to turn it on in an inducible manner? Do you need to be shut off? Do you worry about it being leaky expression? All these things can cause problems. Are you thinking about the untranslated regions? Are there introns of concern? Are there exons of concern? Is there alternative splicing? All these things can mess things up when you start working in eukaryotes and you start asking them to build things for you, okay? Origins of replication. If you're working with multiple plasmids, like in the situation of the suppressor screen, I drew two plasmids, okay? Plasmid 1 and plasmid 2. Are those plasmids compatible? Can they be in the same cell? Do you have selectable markers that allow that? Do you have origins that allow that, et cetera? Are they going to be inherited? Are you inserting something into the germline? Or are you just inserting something into the somatic cell and it's not going to be inherited? Do you need to install tags? Do you need to install affinity tags so you can pull it down from the organism? Do you need to install GFP tags so you can see where it is or localize it? Do you need to install epitope tags so you can do a Western blot, figure out, or immunofluorescence like a flag tag? Is it codon optimized? So if you're taking a gene from, say, Wolbachia and you're putting it in a fruit fly and you're asking the fruit fly to translate the codons of Wolbachia, which is a bacteria, completely different organism, it'll still be able to read the codons, but it might not read them in an optimal fashion. And you might get a very tiny amount of expression. So you might want to codon optimize it for expression in Drosophila. Okay, so this is everything you need to consider. That's 50 minutes of me ranting. I know people have questions on recombinant protein expression. I want to answer those in person in the next lecture. Okay, so let me stop recording here.